which is um, which is they can bind to these molecules, and these, as I said, they are very, very important for drug binding. And as I said, the drug binding is plays major role of half-life and pharmacokinetics and pharmacodynamics. So, I mean, there are many, not only these one, but there are some of this was a old story, but there are again another uh, dozens or not dozens of couples of at least another 30, 40 molecules are been already structurally they're derived all these molecules. And in our study, we have taken several phytocompounds having significant biological functions, including Professor Sangali Muthu, who is also collaborating with our laboratory, and his molecules also is there. The one of the molecule here, he worked on a piperin. Uh, I think I didn't mention here. I mean, we did a lot of other molecules. Some of the data I'm presenting here, you can see this one. So what the phytochemicals it does, you all know that I don't need to. They're all extracted from the plant, and they act as anti-cancer, antioxidant, and anti-inflammatory, and anti-hyperglycemic, microbial, antifungal, anti-arthritic, and asthmatic, and all. So I mean to say that it can cure several diseases, but all the phytochemicals. But any drugs, most of the drugs are derived from the plants only. But these phytochemicals may be less side effects. So we can explore these studies using our all these one. Cytotox study is the first one we said. I'm not going to discuss each and every um, molecule of the cytotoxicity. I just specifically tell one or two molecules. So that will be a boring, repeated the story. So cytotoxicity is mostly, if you take any kind of cell lines, and whether you see that, and whether cell lines, if it is a model of infected cell lines, so whether they can be inhibited the infected cell lines. That is what say that. So this is the macrophage. Mostly macrophages are is usually general the pain management or any kind of a macrophage expresses any diseases. So this is one of the collagen molecules and piperin. You can see here they have antioxidant activities, and we also coumarin derivative. They have good antioxidant. They're inhibiting the IC50 values are about within the range of the FDA. And we also have used some other cell lines, HeLa cell lines, and Ra cell lines. You can see that MCF cell lines. All these diseases. One is this is the liver diseases. This is a breast cancer, and this is the overall. We can for many things. So they have good the even stigmasterol. They have good the IC50 values, where the menthol you can see that. So which can be can candidates to study the pharmacokinetics and pharmacodynamic studies later. People can take into these consideration. One can work on these tools to develop the therapeutic molecules. Also, we did the apoptosis study. Here you can see here a very clear that when you add, you can see that the cell lines. Here, it is live cells are becoming more and more. That shows that this embellin have a significant inhibition of you can see that inhibition very significant, which is be used as a HeLa cell lines. This is one of the cell lines. Um, okay, now these are some of the studies where we have studied all cell lines work. You can see their IC50 values. These are IC50 values at the very beginning. One of the preliminary tests understand the disease model. So we have maybe some of the molecules you can see that andrographite. It is everywhere. They can also will be Hep G liver and the pancreatic or the breast cancers. It can be controlled. But some of the molecules they are very specific to a specific disease. For example, bacosin. Bacosin is very specific to MCF. Is mostly the breast cancer. And you can see that the coumarin derivative. It is specific to only the raw cell lines. Okay, now in order, I said that pharmacokinetics is the one how the drug is acting. So we wanted to study with beginning with the fluorescence studies, the binding one of the fluorometry using fluorometry. We can calculate the binding association and dissociation and also the free energy, which can be, there is a already literature, existing literature is that basing on the literature we can have the values and so like the concentration of the fluorescence. When you add drug, there will be a quenching. With a quenching, these difference can be calculated using binding constant and free energy. Free energy mostly based on the entropy difference between entropy and enthalpy, that is a delta G, which will be calculated. So what we did, this is the equation we use for calculation of the binding association. Then this, and also we did a lot of you can say that whether it is a static quenching or a dynamic quenching. So most of the molecules are static quenching. So if it is dynamic, then it is very difficult to dissociate from the protein. That's why static quenching is more better. So here we did all these one, the binding association. You can see here, the 10 to the power of 4, or this is, it is already literature shows that the 10 to the power of 3 to 20 to the power of 6 is the consideration of FDA approved ones. So if this is in this within the range, we can consider this is a good binding, and also further studies are required. And this, we are not don't want to conclude this molecule can be used for specific disease, but this is only the report. Yes, this can be continued for further if you want to do something, whether how it is acting. You have to have in vivo and in vitro, and in vitro is there. Further you go to animal model, then it go to the clinical trials. It's a long way to go. So this is only a beginning of the study of those phytochemicals. And they're all, they said we should within this range is called a static quenching, and this is the one. And we also have whether these molecules are specifically binding to the pocket, which pocket. We have used a molecular displacement using a known drug molecules. We showed that these molecules can bind to specifically to the one with the pocket. So this one of the molecule is collagen. Collagen is binding to this is replaced by phenylbutazone. That is a marker for a second A domain. So we conclude that this binds to second A domain. We have used several other studies like an atomic force microscopy to see that how their morphology changes when drug is binding. You can see this graph here. You can follow the cursor where this is the control. I mean HSA, when we add incubate with the drug molecules, it forms a kind of the cloud kind of molecules. It's not a bulge kind of molecule. It's surrounded by the human serum molecule. So that's why you can see that wide range, but it means it binds. Maybe several molecule binds, but how they are binding, which have been shown here in the left panel. We also used SPR. I will discuss a little bit later. Use the SPR, surface plasma resonance, to calculate the binding, reconfirm with the fluorescence. So they're all under the level of, this is the dissociation constant, which is agreement with the previous fluorescence studies. We also did mass spectrometry to show the binding, how many molecules are binding to each pocket. So if you see that HSA is a molecular weight of, is the Daltons. And when you have a embelin molecule, this is about 294. You can recalculate 563 to 294. It is almost approximate, not approximately, exactly binding to 1-to-1 range and to that one. We have published all those molecules. So it binds one pocket to one molecule to one molecule. We can see this. So further, these are the one table showing all these, the binding constants free energies and the number of binding sites. So all of them are 1 to 1, and also you can see all this one binding range. I said 10 to the power 3 to 10 to the power 6. They're all binding within the range. Next, going to the structure, when protein is binding to the drug or drug is binding to the protein, there will be a structural changes, conformational changes. You look at this one, protein diamond, 2-dimensional Ramachandran plot and pi and psi angles, how they will be moving, when molecule is binding, they will stretch like a bending, and then little kind of, conformational changes will occur. Looking at, the CD structure, you can calculate alpha helix, beta sheets and random coils of the secondary structure. Then basing on that, we'll say that how much protein conformation is happening. That's what we have done here with one of the molecule, one of the phytochemical molecules. You can see here, nice. They are all the CD is slightly changed. When you can see, another molecule, amylin molecules have a more significant change of protein, but whereas this is based on the temperature we also did, but when you have use a different temperature, whether the binding is dissociated or what, that's what we checked. But here you can see here, you can follow the cursor with the lower it going down. That means minus scale, where there is a slight changes about 6 to 7% of alpha helix. Beta sheets have been decreased, but random coils has been increased, which is an indication that there is a structural changes. That means conformational changes. We also have done the electron microscope, where it forms as similar to our the atomic AFM. This each one is one HSA. Whereas when you see here, there is slightly increased the when you see this one, it is like a forming like aggregates. They form aggregates. That means protein, and I said in IFM similar to that one. They form a kind of aggregates, but how they are aggregated are very strong or loosely, that we have to see with a little more a single particle analysis. That it's a limitation. So whether they are tightly bound, but there are many molecules binding, but only the 1-to-1, maybe some molecules only binding, but rest of them will be like kind of getting precipitated with the HSA molecule. And these are some conformational. You can see with several molecules how they are, all of them, most of them, within the range of conformational change. There is a range. It should not cross more than 20% of conformation. So all of them are within the range. So that means all these candidates can be used as drug molecules, but further studies are required. So to conclude this chapter, you can see that cytotoxic studies with the different cell lines indicate that they have a good cytotoxic effect, which is evident from IC50 values. And as I said, there are all these binding, in vitro binding studies are within the range of 10 to the power 3 to 10 to the power 6 molar, which is in a agreement with the FDA approval. And it indicate that bioactive compounds are strong enough to bind with HSA, which may increase the half-life and efficacy for the therapeutic action if this is within this range. And then there also, I said, the protein changes are there, but they are not that significant. But it should not change that much, and which is an indication that there is an unfolding of HSA of interaction that influence the structural changes of HSA. And binding studies of bioactive that we are not going to talk. So now we did a lot of the docking studies. Maybe most of you may be knowing, aware of this one. This is also this one. Can any of you tell who got the Nobel Prize on this? Okay, anyway, so it is here, discover novel, the drug molecules. This is the method. The binding is very important. I mean, these revolutionary, like the computational biology, given a revolution that all of us know that if you want to release a drug molecule to the market, it takes at least 20 years, a real authentic molecules. But because of the computational study, there is about half of the time can be saved. And a lot of all in vitro studies, strictly in vitro studies, one can understand there are several companies, pharma companies working on. Every pharma company is now recruiting the real bioinformatician who can work on those drug binding studies. So what we did here, we have taken several confirmations, 30 confirmations. Then we have taken with the experimental value, we have taken experimental value close to experimental value, that is our confirmation, we have calculated. Vice versa, both, if it is fluorescence is not, not showing, that means if there is something discrepancies, there is something, maybe may not be a good candidate for, drug candidates, because if you are giving different kind of results, maybe not the stable compound. What we did here, and I said binding, several molecules, they bind, 1A domain, second A domain, third A domain, and the third A, third B, and second A and second B, and first A generally don't bind. First B, there are several molecules binds to the drug molecules. So we calculated all these one, one can understand all the looking at this one, we can do reconfirmation with a lot of mutation studies. That's what we did in the end. And we can manipulate all these one, whether this is true. These are all the real, in vitro studies where, can manipulate all these one, in the, using E. coli model, you express them or, mutate them, these HS and express them in E. coli and see that whether this is real binding or not. That's, we did several studies like that. So these are all some binding studies. So you can see here the binding constants of all these one, the binding constants we have also, you can see here. In silico, the one which are docking, and the in vitro, that is the one fluorescence, and also the SPR studies. And they're almost a similar kind of studies. And free energy is almost same with one. And you can see that not every molecule will not bind to the same pocket. That's what you can see, that most of them are binding to the second A or, and third B, third A also. But mostly second A is the one major binding sites for several molecules. And another very important is the molecular dynamic studies, where, you can see or read that in all these one, these are the importance. The dynamic studies are the more appropriate to understand the molecular binding studies. So what we did here, you can see here RMSD, radius gyration, or RMSF, we have calculated from the simulations. So we did about 1,000 picoseconds, 10,000 picoseconds, which is about 100 nanoseconds, where the molecules are, you can see like this. You can get like this, when it binding in the computationally, they have a kind of a structural stability. That stability will be telling about how they will be, how much time it required to bind to the protein, that, and also what is the free energies and other, stability will tell about. These the red values are the one we tell about. But whatever one molecules we have done, they're all perfectly binding, and we also say that, you can also identify exactly the binding pocket, whether whatever the docking, whatever the fluorescence, what we have done, is it correct, or, to validate this one, this is the best way to do that. Okay, so you can see that, docking reveals the binding of, they have various kind of bindings, hydrogen bonds or hydrophobic interaction. They play a major role for stabilizing the drug and protein interactions. And also, they bind various pockets like second A and third A. And dynamic studies will tell about, indicate the consistent stability of HS biotic compounds. Okay, then this is very important, most of you, that, as I said, the binding efficiency of uremic toxins. This is the very, uremic toxins are, basically a protein, the proteinuria, you can also say that. Protein-bound uremic toxins are that is a very crucial, remove from the dialysis, or, the kidney will be, give it troubles if you don't remove the protein-bound uremic toxins. Most of the chronic patients or kidney patients, it stays with the kidneys and in the body So where, it creates a lot of, side effects. Ultimately, the damage is the kidney. Thing. So what we have studied here, I said, the extraocular liver support, liver support devices albumin as a dialysis. So now these main albumin, the center of the three domains, which is one, it can go and bind to the, to the a kind of a frame. You have like a tube, and the albumin will be attached to that one, like a chip kind of thing. And then when you flow through the, whatever blood will be connected to these tubes, and all these bad molecules bind to HSA. So then you can remove later. It's like a column, the same thing like a column chromatography. So that's what it does. But here, to improve the efficiency of that one, what we did, we have separated, cleave all these products. Like we have expressed individual the domains. That's domain A, domain 1, and domain 2, domain 3, and also we swap like a combination of these domains. We express them in the, like this D1, D2, D3, 1, 2, 2, 2, 3. These are the swabbing. And this one we cloned them. These are the uremic toxins. So we have cloned them in Fissia. And finally, these are the all these expression profile and the purification profile. Then we did all these binding studies by using SPR studies. So we got a nice, took long while to do all these, the expression and purification. We have purified all these domains. You can see that domains are here, D1, D2, D3, D1, D3, D2, and HSA expressed ones. And we characterize by using mass spectrometry. We have identified whatever we have done, all these studies are good. So I will not talk about this because it's only the confirmation how these domains, how they will be stable. So we used the SPR, I said, surface plasma resonance, one of the authentic technique to study drug binding, protein-protein interaction, and drug-drug interaction, and protein-drug interaction. Any ligand, you can study with this. So it will be a curve like that. When it goes, control, when it is binding, you get a, either increasing or decreasing cost. You can calculate association, equilibrium, and dissociation constant using this curve. What we did here, you can see here, all this one with the various concentration. When you add to the domain which we expressed, we did with all these uremic toxins, and they are binding very nicely to individually. They're binding to individual in order to bind to the the total pocket. That's the very important of this study. So what we did here, you can see all these binding. Maybe I will go with these studies here. The summary here. These are all the uremic toxins. So if you can see here, so now HSA, some of them are, these are the one, binding. But if you can see here, the CMF, they're all binding. You can see here, and all these molecules, HSA, IAA, melatonin, CMFF, all these molecules, how they bind, you can see in the down here, D3, most of them you can see that. First, D3 is more favorable for this, HA1. And, even IAA, D3 is good. Melatonin, D3 is good. And CF, this is the one, if you can make dialysis unit with this individually probed with these, the domains, you can purify several molecules so that, you can eliminate these toxicants, uremic toxicant, so that dialysis will be much better. This is now we are going to patent this one. To conclude this one, the affinities of domains are significantly different from HSA. As I said, all this one, the uremic toxins, they bind differentially to the different domains. Domain 3 is showing high affinity, which I told you just before the last slide, and to the hippuric acid and indolactic acid and melatonin. Domain T is showing high affinity to 3-carboxylic methyl and 5-propenyl, this one, CMFF. among all these no the protein molecules. I mean, whatever the melatonin has a less affinity to HSA and its domain, except melatonin. All other molecules are very good in binding to all these domains. So now, what we are concluding here? The combination of basic and aliphatic amino acids are forming favorable binding condition for the ligands in the binding sites, providing the way to improve the efficacy of dialysis in extrapleural dialysis. Actually, there are many companies come forward to collaborate with us in this study. There are several companies like Verco and then also Bharat Biotech. They wanted to have our technology. So finally, I would like to conclude here. You can see here nicely whatever the molecules you can see. We have done all these phytochemicals where they are binding. Some of the molecules, only two we have identified, 1B, and most of them we can see the secondary domain, and several of them are binding to third domain. There are very few binding to third domain. That way, it gives a lot of information for the pharmacokinetic studies, and then further studies has to be carried out to understand their drug behavior. Studies are can be as I said, can go in vitro and in vivo and the clinical study. Okay, and so here I anyway all these one because without funding I cannot we cannot do anything. All these one funding bodies. We have national and international collaborations, international collaborations, and the students who done all this work. Aparna, Shreya, and the Mahesh. These are the people who have contributed for the study, and all these the postdocs also. We have Veerababu and Manjunath who worked on this. Thank you for listening, and if you have any questions, be happy to answer questions. And thank you for giving me the opportunity. Yes.
Yes, sir. Happy to see you, sir, here.
Oh, Professor Sangali Muthu, yes.
Yes, sir. After long time, we have been able to visual work. Thank you for your reference. Thank you for your talk, sir. It's very useful for us. We have enjoyed a lot. Sir, few more questions from audience, sir. So you used atomic force microscope, AFM, for analyzing the binding studies. So, can you explain how this technique are used to analyze the binding property of small molecule or different molecules?
Yeah. The AFM used generally to see the morphology of protein. The protein structure. Actually, this is a very basic one what we have done. So we have to do a little bit more of images and image analysis to understand more of the structural analysis can be done. But what we showed here is only how they change. Whether as I said, some of them the studies. For example, mass spectrometry, where we have seen one-to-one interaction, and the binding can be one drug binding. Let's say a secondary domain, or secondary domain. Most of the proteins are bound to secondary domain, and we wanted to understand really secondary domain, or it can form all entire over the protein. So that's what we say that what we have seen that it is not really only specific to that one. They are loosely bound to other binding pockets. So that can understand, it can bind to overall, entire protein. Is it clear?
Yes, sir, it's like here. So you have, left my molecule of that list. I had to. You have given the list of molecules on binding of most molecules, right? Okay.
Actually, I'm not able to hear actually.
Okay, sir. So you have provided a list, no, sir, the phytomolecule having a good binding ability on Neosera molecule. Can you hear, sir?
No, I very low voice, very low voice actually. Actually, very low voice. Yeah. Can you repeat? I know because I'm not able to hear the question.
Yes, sir. Yes, sir. Now, you are hearing, sir, my voice? So can you repeat?
Sorry. Hello.
Yes, sir. Sir, one more final question, sir. The binding range of molecule, you have given the binding knowledge. Binding knowledge